Welcome to the lesson on the Intro to Mitosis. Sorry for the delay. Slides were not moving. Welcome to the lesson on the intro of mitosis. Here we're going to talk about the purpose of mitosis, and also interphase and what happens during interphase. Interphase is technically not considered part of mitosis. It is the stage before mitosis occurs, in which the cell prepares to divide or lives its normal life. So, you all began as an egg cell that is smaller than the period at the end of a sentence. And to become you, that cell divided thousands of times, maybe even hundreds of thousands of times, by mitosis, okay, to create all of your cells. Okay, those cells then had to undergo a differentiation process to become the different cell types, which we're going to talk about later in the course. But the point is of this slide is that you all started off as a single cell, which had to divide thousands and thousands of times, okay, by the process of mitosis. So if we look at this pictures here, this is the egg dividing into two, into four, and then into many cells. When a cell divides without growing, it's called cleavage. And this here is a blastula, or a ball of cells, that happens during embryonic development. This will continue to divide and then eventually undergo differentiation, where it becomes different cell types. But in this unit, we're just focusing on the process of mitosis. So cells divide for the purposes of reproduction. So there are some cells that reproduce asexually. These are usually one-celled organisms, and all they do to divide is do mitosis. In asexual reproduction, because we're doing mitosis, the parent is identical to the daughter cell that's produced, where the two cells produced are identical. We also use it for growth. So obviously when the egg is in the embryo, it divides and grows to create a multicellular organism. And the last reason why we do mitosis is for repair or renewal. If you ever get a cut in your skin cells divide to replace the skin cells that were injured. Some organisms have the ability also to regrow their tail if it gets cut off. Cells that are damaged in many places, in many organs, can regenerate itself. For example, the cells of your stomach regenerate themselves almost about once a week. Also, the cells in your mouth also regenerate often. So whenever cells die, which is happening all the time, the cells around them divide to repair or replace the cells that were injured or died. So again, the three reasons why cells divide is for reproduction, for growth, and then to repair damaged cells. So the cell spends about 90% of its life cycle doing its everyday job, okay? Producing RNA, making proteins, whatever the job of the cell is, there are different cells that have different jobs in the cell. If triggered, though, it will prepare to divide. So 90% or most of the life of the cell is spent in G1, okay? Where the cell is living its normal life, doing its everyday job. If it's a red blood cell, it's carrying oxygen. If it's a muscle cell, it's making ATP to allow for muscle contractions, but whatever its normal job. But at some point, the cell may get a signal to divide, and then it will enter S phase in G2 and complete mitosis. Okay, we're going to talk about that signal to divide next week, okay? But today we're going to talk about interphase and what happens in interphase. In G1, again, the cell is living its normal life, doing its everyday job, and it may leave G1 and if it gets the signal to divide. So if we look back here, and so we said in here, what we're writing on this slide, what we said in G1 is that the cell lives its normal life. So we should be writing this on this slide. Whatever the job of the cell is what it does in G1. If it's to carry oxygen, if it's a muscle cell to contract, if it's a liver cell produce many enzymes. So whatever its job is, it does during G1. Okay, it then may enter S. Okay, and if it gets the signal to divide, it will enter S, and that's where DNA replication occurs. So the DNA in the nucleus will double. And we're going to talk about how DNA replicates in December. And then after it replicates its DNA, it's going to enter G2. And during G2, sort of the last growth phase, okay, in G2, the cell grows, gets a little bit bigger, and replicates its organelles, particularly the mitochondria, some of the ER, more organelles for the new cell that's going to be created by mitosis. So those are the three phases of interphase. G1, cell lives normal life. If it gets the signal to divide, it will enter S, where it's going to replicate its DNA. And then after it replicates its DNA, it enters G2, where it grows some more and replicates its organelles. We're going to see that there are checkpoints to check to ensure that this process, all these processes that need to occur in S and G2 happen correctly. So cells sort of have two paths. So let's say you have a cell that's made by mitosis. It can grow, live its normal life, and divide. And this happens in epithelial cells, those are skin cells, blood cells, stem cells, which become any type of cell. These are cells that divide often. Okay. Or the cell can enter a phase known as G0. And G0 is when once the cell is created, it never dies. Okay. Brain and nerve cells, once they're created, they don't reproduce, and neither do muscle cells. So if you injure or kill muscle cells, a lot of times they actually have to amputate because they don't want blood going to an area of the muscle that's not working. So muscle can't regenerate, and brain cells can't regenerate, or they can't redivide. Liver cells spend most of their time in G0, but then if there's liver damage, they can re-enter the cell cycle. But it often happens slowly, and that's why alcoholics can get cirrhosis, which is when you have damaged liver cells, which take a long time to repair because they spend most of their time in G0. They're not naturally reproducing. But if there is enough damage, they can re-enter G1. But it does take a while for liver cells to repair themselves. So again, we have two paths: a cell that is continuously dividing, or cells that exit the cell cycle and enter G0. And those are brain and nerve cells. Liver cells enter G0, but then can re-enter G1 and go through a cell cycle if severe damage occurs. Okay, so now we're here to the goal of mitosis. Okay, and the goal of mitosis is to create identical cells. So that's what we should write on this slide. The goal of mitosis is to create identical cells. To do that, okay, we need to, remember we replicated our DNA in S of interphase. So to create identical cells, we need to separate the chromosomes evenly. And that is basically the whole point of mitosis, is to create identical cells by separating the chromosomes evenly. Okay, and that's going to be the point. So when you watch the videos that show the stages of mitosis, the purpose of mitosis is to create cells that are identical, and to do that, it has to separate the chromosomes evenly. And that's what mitosis accomplishes. So for tomorrow, because you're probably watching this on Sunday, we're going to have a quiz on the stages of mitosis and what happens during each one. I will see you on Monday.